Welcome and hello everyone. Thank you for joining us for our live MDA Engage ALS Symposium. My name is Nicole Petrowski. I'm MDA's Community Education Specialist. We appreciate you joining us today and we're glad to be able to share this important symposium with you. I especially want to thank our supporters for this event, Alexion Pharmaceuticals, Amylyx Pharmaceuticals, Apellis Pharmaceuticals, Biogen, and Cytokinetics, and welcome any of their representatives who may be joining us today. We really appreciate your support. MDA is committed to community education, and we believe in bringing our community together for opportunities to learn from specialists and having opportunities to connect with others. This event is part of a larger MDA Engage series with disease-specific symposia, which we are hosting today, community education seminars, and community webinars as well. I wanted to share with you our schedule for our upcoming community education seminars that we'll be hosting throughout the end of the year. These are day-long educational seminars, and they provide individuals and families the knowledge and resources around neuromuscular disease. Information is shared by experts in the field on a variety of topics related to care, research, and resources. And these are our community webinars we have lined up through the summer. Families can learn about different topic areas, and these are typically only an hour long. So each webinar will feature experts in the field, along with opportunities to ask questions from the presenters. And here's a look at our agenda we have for today. As you can see, we have quite a busy afternoon planned. But before we begin, I wanted to go over a few housekeeping items. First and foremost, we get this asked a lot. We are recording today's event, and we will be posting it to our mda.org website for on-demand viewing in a few weeks. For those of you joining the event live, please know all phone lines have been muted. We will be having Q&A session at the end of each presentation, so please utilize the Q&A icon to type in your questions. If you hover over the bottom of your screen, a tray of icons will appear, so just click on the Q&A icon to open that feature, and make sure you enter your question to host, and you don't need to wait until the presentation is over before submitting questions. Feel free to use the chat feature during this seminar as well. Make sure you click on all panelists and attendees when you type in your comment. And then you will notice on the agenda there are a few small breaks between speakers. Please feel free to stay connected to the live broadcast during these times. This is the time when I will be checking audio for the next presenter, so you will hear us speaking. And then finally, I will be sending out a brief survey after our symposium today and tomorrow actually, and we would like to receive your feedback on what you heard. We want to make sure we are discussing topics that are of interest, and we always use your feedback as a way to improve future educational events. We also will be doing a drawing for 2 winners for a $20 Amazon e-gift card who submit a survey. So thank you in advance for that. I also wanted to share with you tomorrow's agenda in case you haven't registered for that. We will be hearing from some wonderful presenters who will be speaking on some very specific topics surrounding ALS. So if you haven't registered for tomorrow, please visit our mda.org page, and under the Care and Services tab, click on Community Events, and you will find our disease symposia listed there. Now I would like to kick off our symposium by welcoming our keynote presenter, Dr. Richard Bedlack. I know many of you are very familiar with his work. Dr. Bedlack grew up in a small town in central Connecticut. He went to college at William & Mary in Virginia, and then back to Connecticut for an MD and PhD in neuroscience at UConn. Finally, he came to Duke, where he completed his medicine internship, neurology residency, neuromuscular fellowship, and master's in clinical research science. He is currently a professor of neurology at Duke and director of the Duke ALS Clinic. He has won awards for teaching and patient care, including Best Neurology Teacher at Duke, Healthcare Hero, Strength, Hope, and Caring Award, America's Best Doctor, the American Academy of Neurology Patient Advocate of the Year, and the Rasmussen ALS Patient Advocate of the Year. He has received ALS research grants, participated in ALS clinical trials, published more than 120 ALS articles. He is a leader of the International ALS Untangled program, which utilizes social networking to investigate alternative and off-label treatment options for patients with ALS, and leader of the ALS Reversals program, which attempts to understand why some people with ALS recover from it and to make this happen more often. He lives in Durham, North Carolina with his wife, Shelley, and his 2 mischievous cats. So thank you for being here, Dr. Bedlack. I will go ahead and turn it over to you.
Well, thank you so much for that nice introduction, Nicole. Let me just pull up my slideshow here. Hello, everyone. Thanks for joining me today. I'm going to give you an update on what I think are some of the most exciting things happening in ALS research right now in May of 2021. And I call this talk Signs of Progress and Reasons for Hope. I'll start off just with some disclosures. I do have research support from some groups, ALS Association, Healy Center, Medicine Nova, and Orion. And I do have consulting income from Alexion, the ALS Association, Amylyx, Biogen, Brainstorm, Mallinckrodt, MT Pharma, Nubiotic, Orphazyme, and Woolsey Pharma. So these are the topics that I want to cover in my hour today. We'll talk very briefly about the evolution of supportive care, which is a really important part of what we offer people with ALS now. We'll talk about how we've been and continue to target what I call the downstream pathways that result in the death of motor neurons in this disease. And then we'll turn our attention to what I think is the most exciting thing going on right now, which is the targeting of causes of ALS for the first time ever. We'll talk a little bit about understanding ALS resistance and what I call ALS reversals. We'll talk about improving patient engagement in research and improving our trial designs. We'll talk about expanding access, a really hot topic in ALS right now. And then finally, I'm going to tell you about something new that I'm personally very excited about, which I think is a form of high throughput screening in people with ALS. And so let me first tell you about my first experience with this disease. It was 24 years ago. I was a resident here at Duke, and you can tell that this picture dates from the late 90s because that's the only time in my life when I wore blue blazers. I was trying not to make too many waves here when I was a resident, and also because I had the Jerry Seinfeld hairstyle, which was very important back then. But I will never forget that person's story or the amazing collection of findings on their physical exam. And I'll also never forget how horrified I felt when the attending that I was learning from came in and said to the patient, well, we know what this is called, but we really have no idea why it happens, and there's nothing we can do about it. You just have to go home and get your affairs in order. And that didn't sit well with me then, and it still doesn't. And I think as I was driving home that day, that's when the seed was planted for me to stay at Duke and to build my ALS program, which offers options and hope to people who come here. And I'm so pleased to say that things are a lot different now in 2021 than they were. There are entire books written about all the options that can be offered to people with ALS, including multidisciplinary team care, nutritional interventions, respiratory interventions, exercises, equipment, and symptom management. And, you know, some people look at these and they say it's not much, but I can tell you firsthand from having seen 3,000 patients in the last 21 years that these things add up and they make a difference. They improve the quality of life for people living with this disease. They reduce hospitalizations, and together they add up to significantly longer survival for people with ALS now than in the late 90s. So we've also learned some things about why motor neurons die in ALS. We know there are at least 10 different downstream pathways that are involved in this. So there's alterations in what's called nucleocytoplasmic transport of RNA molecules and RNA binding proteins. There's alterations in RNA metabolism with several important RNA binding proteins mislocalized. There's impairments in something called proteostasis with accumulation of aggregating or clumped proteins. There's impairments in DNA repair. There's mitochondrial dysfunction and oxidative stress. There's oligodendrocyte dysfunction, which leads to reduced trophic support for motor neurons. There's neuroinflammation with activated astrocytes and microglia, secreting fewer neuroprotective factors and more toxic factors. There's defective axonal transport, defective vesicular transport, and finally something called excitotoxicity, which occurs because of the loss of a transporter in the membrane of astrocytes. And that results in an excess amount of a chemical called glutamate, which is poisonous to motor neurons at too high a level. So we've started to address these downstream mechanisms. The medication that we first learned was effective in ALS is called Riluzole or Rilutec. This is an oral glutamate antagonist as well as a membrane stabilizer. And it's very clear, just about every study that's ever been done has shown that people who take Riluzole live longer with ALS than people who do not. So it's quite clear that that works. In my experience, it's also pretty well tolerated. It's pretty easy to monitor for the one serious side effect of Riluzole, which is elevations in liver transaminases. The other medication that's more recently approved is called edaravone or Radicava. This one is a little bit more controversial. It clearly has one study showing that people who took this medication progressed more slowly on a measure of disability called the ALS Functional Rating Scale compared to people on placebo. But this was a very highly selected study. These were people who had ALS for less than 2 years, who had lots of preserved function on the scale that was being measured, as well as on breathing measurements, and who were progressing very quickly. Another study of people who were a little further along in the disease was unable to show a benefit. Most recently, we learned about a new medication which may soon be approved in certain countries, and that is called AMX 0035. This is a combination of two products. First, there's a histone deacetylase inhibitor, which is called sodium phenylbutyrate, and then there's also an antioxidant, which is called TUDCA. And what we learned about this over the past year is that when added to Riluzole, this may additionally slow the progression of the disease as measured by the ALS FRSR by about 40%, and it may additionally prolong survival by at least 6 months. And there are many other treatments with different mechanisms of action that are currently under study in the ALS space. And I think over the next few years, we're going to be prescribing cocktail therapy for patients with ALS. So we will be giving people a combination of medications that have different mechanisms of action that affect different of those downstream pathways. And so some of the pathways that are currently being targeted include complement, microglia, neuroinflammation. There's membrane stabilizers that are under study, newer antioxidants, energy boosters, growth factor delivery systems, and even drugs that target muscle, like troponin activators. So there's a lot going on that we might soon have the ability to prescribe in the clinic. And there's a couple of very new things that I wanted to mention today that many of you might not have heard of, because these are studies that I'm mainly doing here at Duke. The first of these is a drug called Clinbuterol. This is a beta agonist. It's used to treat asthma in many countries around the world, but it's not FDA approved for any indication. Many American sports fans have heard of this because there have been several high-profile athletes that were suspended from their sport for testing positive for CLEN, as it's sometimes abbreviated. Now, why do they use it? Because it actually stimulates muscle growth by stimulating something called insulin-like growth factor 1. That's cheating in sports, but it's exactly what we want to happen in a neuromuscular disease like ALS. It has been around in the ALS space for at least 15 years. It was shown to improve motor function and survival in an ALS mouse model. And it was even in a small uncontrolled study. They gave 16 patients with ALS 20 micrograms 3 times a day, and they just watched what happened over 6 months. Again, there was no control group in this study. But here's a table of some of the main outcome measures in this pilot trial. And what I want to show you is that the muscle strength scores, the average muscle strength scores in the upper limbs and the lower limbs were significantly higher at the end of 6 months than they were at enrollment. Also, the forced vital capacity was significantly higher after 6 months of treatment than it was at enrollment. one thing that's not shown here is the ALS functional rating scale was stable over 6 months on average in this group of patients. In my experience, it's very unusual for a group of patients together in a study to show improvements like this just spontaneously. So I decided to get together with someone at my institution named Dr. Dwight Coble. He's a pediatric neurologist who has a lot of experience using Clinbuterol in kids with muscular dystrophy. And together we decided to do another pilot trial. And like every trial that I designed, I tried to make this as inclusive as possible. I decided there would be 25 people with ALS. Everyone would get the Clinbuterol. And Dwight thought we could taper up the dose to be much higher than what was used in that previous trial. He thought we might even be able to do better than they did. And so we tried to target 80 micrograms twice a day for 6 months. Our main outcome measure was the slope of the ALS functional rating scale during treatment compared to the slope before treatment started. And we've got some other outcomes: forced vital capacity, muscle strength testing, and safety labs. I also tried to make this as easy as possible for those who got in to complete. There were only 4 in-person visits over 6 months. The rest of the visits were telephone. And our last patient completed this study in February of 2021. And so what I'm about to show you is hot off the presses. It's what I would call very preliminary. The first thing we know so far is that the dose we chose was not nearly as well tolerated as the smaller dose in that previous study. Only 11 of our 25 patients completed the entire 6-month study. The main reason for dropout was side effects: cramps, tremors, heart palpitations, insomnia. These are all known side effects of this class of drugs. But I will say this: the 11 people who stuck with us had pretty interesting findings. Every single one of them had a slowing in their ALS FRSR progression during treatment compared to how they were doing before. And 5 of the 11 actually had slopes on treatment of greater than or equal to 0, meaning they were either stable or they were recovering lost functions. If you look at this group as a whole, collectively, we showed a 70% slowing in the rate of ALS FRSR progression while on drug compared to before the trial. Now, of course, this trial is far from perfect. It's very small. It's not randomized. It's not placebo-controlled. And so far, we've only analyzed the folks who finished the study. We have a lot more work to do, including analyzing everyone in what's called an intention to treat analysis, as well as analyzing all the outcomes. But to me, even the data that we have so far, when placed on top of the other pilot study and the animal study, suggests that we need to move this further into a larger randomized trial soon. Another project that's targeting a different downstream pathway that's unique to Duke is called Neuropheresis. So one of my colleagues here is a neurosurgeon. He's invented a device that can actually filter spinal fluid. And he's using this device to treat patients who have bleeding in their spinal fluid, something called subarachnoid hemorrhage. Now, what does this have to do with ALS? It turns out that the spinal fluid from people with ALS is toxic. If you dump it on astrocytes or microglia in a dish, they change. They become pro-inflammatory. If you inject the spinal fluid of a person with ALS into a mouse, the mouse develops something that looks like motor neuron disease. What's in there? We don't know for sure. We hypothesize that there are small molecules called cytokines that are involved in driving neuroinflammation. And there's one in particular that we're interested in, which is called chitotriosidase 1, or KITE1. Now, the problem is these are very small, and the filter that we has a pore size of about 100 kilodaltons. And so we don't know whether we can filter these small molecules with the device we have. And so we've designed something which we call a proof of concept study. We want to obtain spinal fluid from 5 people with ALS, and we want to run it through the filter and see what we filter off. In particular, can we filter any of these small molecules? We also want to inject filtered spinal fluid into our mice to see if it still makes the mice sick. And we feel like if we can actually filter off something plausible and that filtered spinal fluid no longer makes mice sick, then maybe next year we can go to the FDA and get permission for a small pilot trial in people. So I do have some very preliminary results from this as well to share with you. So this diagram over here shows where we're injecting the spinal fluid of a person with ALS into a mouse. There's a little area here called the cisterna magna. And this is the assay that I'm going to show you today. Basically, we take a mouse and we put him in a plexiglass box and we put a camera overhead and we watch how much he moves around over a certain period of time. And I'll show you some of the videos. So here's a healthy mouse. Nothing's been done to it. And you can see that he's very interested in exploring this entire plexiglass box. Now here is a mouse that's been injected with the spinal fluid of a person with ALS. And as you can see, that mouse doesn't move around very much. And then finally, let me show you what happens to a mouse that's injected with filtered spinal fluid. Looks a lot more like the healthy mouse. So there is another way that we can analyze these kinds of results. We can actually do traces of where the animal goes. And I think you can see by these traces that the mouse that was injected with spinal fluid from a person with ALS is not doing much compared to the healthy control. But when we filter the spinal fluid before injecting it in, that mouse behaves a lot more like a healthy control. So we're pretty excited about this. That's the only reason I'm presenting it today. It's very preliminary. We need to replicate this a few times. And we still haven't figured out what exactly we're filtering yet. Those results, those experiments are all underway right now. So I mentioned in the beginning that probably the single thing I'm most excited about in ALS is that we are, for the first time in history, targeting causes of the disease, not just downstream pathways, but causes. And as you probably know, the best understood causes of ALS are genetic. About 10% of people have a gene that's broken, that's mutated, that we think is causing their disease. And we call that genetic ALS or familial ALS. As of today, we know of more than 30 different genes that can cause ALS, and we keep discovering more. So this is a timeline of gene discovery in ALS. And as you can see, you know, for the first part of my career, we knew about hardly any of these genes. And it's really over the past 10 years, especially, that we've discovered so many of them. And of all these, the one that we understand the best is the first one we discovered. It's called SOD1. And this causes something called familial ALS type 1. So this accounts for 3% of all cases of ALS. There's 150 different point mutations in the SOD1 gene that can lead to ALS. And we can use this information to build models. And so we have ALS mice and rats and flies and fish and worms. And we can really study what's happening at the cellular level in those kinds of models much more easily than we can study humans. And what we know for sure is that these mutations are causing something called a toxic gain of function. They're resulting in a protein which gains a new function which contributes directly to the death of motor neurons. Now, what exactly is that function? Well, there's still some controversy about that. We know that this mutant protein gets secreted through something called chromogranins, and then it's taken up from the extracellular environment. We know that extracellular misfolded mutant SOD1 can activate microglia by binding to a specific receptor. And after that happens, the microglia become pro-inflammatory. We know that the expression and uptake of misfolded SOD1 causes stress to part of the cell called the endoplasmic reticulum. And that results in the secretion of a protein called CHOP, which causes the cell to go into something called apoptosis, basically causes it to kill itself. And so there's a lot of different things that this is probably doing. But the bottom line is the way we should be treating people with this form of ALS is to try to get rid of as much of this mutant protein as we can. And the simplest way to do that is probably something called an antisense oligonucleotide. So this binds to the mRNA and prevents it from making the mutant protein. And I'm excited to tell you that just this past summer, we saw the results of a very small pilot trial that was done of an antisense oligonucleotide directed against mutant SOD1 called tofersen. So this was 50 people with this type of ALS who were randomized to receive 3 different doses of this drug or a placebo for 12 weeks. So small and short, mainly a safety study. The good news is it did appear to be safe. It did appear to be well tolerated. It did result in a significant reduction in CSF mutant SOD1. But what's really exciting, and I think was unexpected, is that when they looked at the fastest progressing patients in this study, people with certain specific mutations like A4V, who changed very quickly, they were able to show a dramatic slowing in the rate of progression, even over just a 3-month study. And that's what this graph shows you here. This blue line are people who are on placebo over the 3-month study. And you can see that they changed a lot on the ALS functional rating scale. And this red line is people who are on tofersen. And you can see that they changed very little, if at all. And so the next phase of this is now open for business. I said that SOD1 was the gene that we understood the best. It's not the most common gene to cause ALS. The most common gene to cause ALS is called C9orf72. And it's probably somewhere in the neighborhood of 3 times as common as SOD1. This is a hexanucleotide repeat expansion, which occurs on chromosome 9. It was just discovered about a decade ago. There's still some controversy on exactly how this produces disease. Some people think that it might be through a loss of function, and there's still some controversy about exactly what the function of the C9 product is. It probably has to do with RNA transport in and out of the nucleus. But other people think it's a toxic gain of function. And there's sort of 2 schools of thought about what that might be. First, the RNA that also have repeating sequences tend to form something called RNA foci, which are toxic. Second, this RNA can be translated in 2 directions. And when it's translated in a sense direction, it actually results in something called dipeptide repeat proteins. These are small, very toxic proteins that this gene can produce. And so we do have some trials underway targeting C9orf72. So one group of trials are antisense trials that are trying to shut down as much of the product of this gene as they can. And again, those are underway. They're starting off with very small, very short duration safety trials, but they're rolling some of those patients into longer trials where they might be able to get a better idea of the efficacy of the treatment. And there's also a repurposing trial where they're using a diabetes drug called metformin, which surprisingly blocks the production of those RAN proteins, those dipeptide repeat proteins that I showed you. That's open for business down in Florida. So again, the things that we've talked about so far in terms of causes, they're for a specific category of ALS, which is called genetic or familial ALS. Most people don't appear to have that. 90% of people do not appear to have any broken genes causing their disease. We call that sporadic ALS. And interestingly, we are starting to get some ideas on why some subsets of sporadic ALS might happen. There's evidence for an infection, and there's evidence for a toxin that might cause this. So with regard to an infection, the first clue we had that there might be an infectious cause of ALS was the observation all the way back in 2001 that some people who got infected with HIV developed an ALS-like disease. And when these people started treatment for HIV, many times not only did their viral load go down to undetectable, but the ALS-like disease very often stopped or reversed. And so the search was on to find a similar retrovirus that might be able to cause something that looks like ALS. And it's only really been in the past 5 years or so that someone may have found it. It's something called human endogenous retrovirus K, or HERV-K. We all have this retrovirus in our DNA, but it appears that only people with ALS express it in their neurons. Healthy people don't appear to express it. And when you take this retrovirus and you genetically insert it into mice, they develop a motor neuron disease with shortened survival. So this is very exciting. And there's already been one attempt to treat people with ALS with an antiretroviral drug called Triumec. So this was an open label study. 40 people with ALS got Triumec for 24 weeks. One of the main inclusions was they couldn't have HIV, but these folks actually did not require people to have HERV-K expression. And in spite of taking a rather unselected group like this, they did show a more than 20% slowing in disease progression compared to how people were doing before they started the antiretroviral. So there is now enrolling a study at the NIH, which I think has an even better design. If you get into this study, the very first thing they do is check to see if you have HERV-K expression. And if you have it at a very high level, only then do you get treated with antiretrovirals. If you don't have it at a high level, you don't get this type of treatment. I think that's a much smarter way to do the study. And hopefully they'll finish enrollment soon. And 6 months later, we'll know if that dramatically slows progression for the subset of patients whose sporadic ALS might be caused by HERV-K. Another theory about sporadic ALS is that there might be a toxin called beta-methyl amino alanine, BMAA, that causes some cases. This was first identified on the island of Guam, where once upon a time, the risk of ALS was 50 times higher than anywhere else in the world. And there it appeared that this toxin was coming from a seed called cycads, from an animal that the people of Guam ate, a flying fox. And later it was found to be coming from blue-green algae. And when that observation was made, people started to study the brains of folks from outside Guam. And sure enough, in a subset, they do find elevated levels of this toxin. And even more amazingly, they find elevated risks of ALS in people living around bodies of water that are full of blue-green algae. Most recently, I will tell you there's a theory about another source of this toxin. It might actually come from the family of organisms that live in our guts, something called the microbiome, which we're going to talk more about in a few minutes. We think we might understand how this toxin causes a disease like ALS. It seems to get incorporated into proteins in place of an amino acid called serine. And in experimental models of this disease, we can block some of the toxic effects of BMAA by giving the animals lots of the L form of serine. And so that led to the idea of treating people with sporadic ALS with L-serine. And there has been one previous attempt to do this. 20 people with ALS got randomized to one of 4 different doses of this for 6 months. And their progression was compared to a group of people, a group of historical controls. And so here you see the group of historical controls, and you can see that there's a wide range of different progression rates. On average, this group progressed at about 1 point per month. And what you can see in these columns here is that as they ramped up the dose of L-serine, the progression rates became a little tighter, and also they became a little less. And when you got to the highest dose, 15 grams twice a day, there was a very slow progression in these patients. Now, again, this is really small. It's not randomized. It's not placebo-controlled. It's not blinded. But it is very intriguing. And there is now a 50-patient open-label trial open in Dartmouth where they're going to push people up to this high dose of 15 grams twice a day and see how they progress over 6 months. So I'm really looking forward to seeing the results of that. So now let's turn our attention to this topic, ALS resistance and reversals. Everyone listening to this call probably understands that the progression of ALS is very different in different patients. You can look at this in 2 ways. You can look at this by an outcome measure like the ALS Functional Rating Scale. So here's a study from Nature Reviews Neurology that followed, I believe this was 30 people over time and looked at the rate of progression on this scale. And you can see that some people are hardly progressing at all and others are going almost straight down. You can also look at it in terms of survival. And so we hear the statistics all the time that, the average survival from symptom onset is about 3 years with ALS. But, 20% of people live 5 years, 10% of people live 10 years, 5% of people live 20 years. And there's some famous examples of people with ALS who lived, 50 years from the time that they were diagnosed. There are even people with ALS who seem to recover from the disease, which I will talk more about in a minute. Now, this is frustrating for researchers because it creates a lot of noise in our clinical trials. But I personally think this is an important clue. If we could understand why some people seem to be more resistant to this disease, why some people seem to be able to recover from it, we might be able to make everyone more resistant. That certainly has been the case in other diseases. So in HIV, for example, there has long been known to be a group of people called elite controllers who get infected with the virus, but never get sick. And by studying those people, they actually figured out that they have a mutation in a pathway that HIV needs to get into cells, which most people never knew about. And a company called Pfizer was able to build a drug that blocked that pathway that helps everyone with HIV. So I believe this is an important area for us to study. So there really hasn't been a lot of progress in terms of understanding the differences in progression rates between patients. And I personally believe that it might have to do with this thing called the gut microbiome. So as I mentioned before, there are trillions of organisms, fungi, viruses, and bacteria that live in all our GI tracts. There's no question that these organisms play a role in diseases of the gut, like, for example, C. difficile colitis and inflammatory bowel disease. But what's really fascinating is in recent years, there's more and more evidence that this might actually influence degenerative diseases through something called the gut-brain axis. Some of the organisms in our gut produce toxins that can get out and potentially poison cells in our brains. Some of these bacteria produce products like butyrate, which are neuroprotective. Some of these organisms produce things that are pro-inflammatory, like, for example, some of those cytokines I told you about before. So there's been a lot of work in the past 4 or 5 years trying to understand whether the gut microbiome is different in people with ALS compared to healthy controls. Unfortunately, there is no clear consensus on this. The studies are all over the place in terms of what they find. I believe there are many possible explanations for these discordant findings. First of all, there's a lot of differences in the methodology that people are using. They're looking at different types of patients. They're looking at different types of controls. They're getting samples in different ways. Some people are looking at saliva. Some people are looking at stool. They're storing the samples in different ways. They're processing the samples in different ways. They're running different types of analyses. But there's one other possible explanation, and that is that maybe these studies are asking the wrong question. Maybe the gut microbiome does not determine who gets ALS, but maybe it determines how people do with the disease once they get it. And there have been 3 really fascinating recent animal studies that suggest this. So in SOD1 mouse models, in an SOD1 worm model, and in a C9orf72 mouse model, simply by changing the gut microbiome, you can drastically change the rate of progression of the ALS-like disease in these animals. That's stunning. And so there's only been one study that's ever looked at differences in the microbiome in people with ALS who have different progression rates. And this found a clear difference. So they looked at something called the microbiome complexity, also known as the Shannon Index. And they looked at the survival of the patients in their study. And it turned out that the simpler your microbiome, the longer you survived. And it was really quite an extraordinary difference in survival. As you can see, look at the 50% probability of survival. I mean, there's a difference of about 40 months, which is incredible with people with different microbiomes. And so we now are gearing up to look at the microbiome in 3 different types of people with ALS: fast progressors, slow progressors, and ALS reversals. And we're going to look very grossly at it. And then in a subset, we're going to look at, in great detail by culturing the things that are in the microbiome and also by looking at the metabolites. And I do believe this type of research could inform the development of completely new treatments which target the microbiome, such as probiotics. And I hope that these treatments will eventually slow, stop, or reverse ALS. you've heard me use this term ALS reversals a few times. And this is because in the last few years, I have learned that there really are people whose medical records I have in my hand, who really appear to have had ALS, progressed to where they were disabled from it, and then recovered most or all of their lost motor function. As of today, I know about 52 of these from all over the world. And I've got 2 patient-funded programs underway at Duke to try to understand these ALS reversals and to make them happen more often. One is called STAR, Study of ALS Reversals, and there are several collaborations with groups around the world that are helping me to study the genetics of these folks, the RNA, the proteomics, the microbiome, and the environmental exposures. And the other program is called ROAR, Replication of ALS Reversals. This is a program I'm doing where I'm conducting small, very patient-centric pilot trials of some of the supplements that are associated with these ALS reversals. And so the first trial I did a few years ago was of a supplement called Lunasin. Unfortunately, that did not reverse ALS, but I learned a lot about how to make much more patient-centric trials. And I'll talk more about that in a minute. We're currently enrolling in another ROAR trial of a form of curcumin, which is called ferrocurmin here at Duke. So now I'm going to turn our attention to improve patient engagement and trial designs. And so I want to start with patient engagement. So when I first started my career, I built my multidisciplinary clinic, and I got my name out there. We had a very large number of patients. I was highly encouraged and excited about doing research, and we started to bring clinical trials into the clinic. And I realized that only a very small percentage of the people I was following were actually going into these trials. And it led me to do a few surveys of other clinician scientists like me who were doing trials, and also surveys of patients and families. And the net result of those surveys is I learned there was a huge gap. There was a huge problem with patient engagement in our field. I found that many people with ALS did not know about the research options they had. Many of them did not feel like we were asking the right questions. Many of them were sort of turned off by how difficult our studies were to get into, how difficult they were as far as all the things that we were asking, the numbers of visits, the length of the visits. Many of them did not like the fact that we had placebos. Many of them were frustrated by how long it took from the time that we started a study until the time that we got a result, in some cases years. And so I looked around and I found that other fields had a program where they were training people with those diseases and their families to understand research better, and then giving them a regular forum to interact with different stakeholders in their disease spaces so that they could influence the way research was being done. And so I decided to try to replicate these programs, which are called Clinical Research Learning Institutes. And through these, we've now trained more than 400 people affected by ALS to understand research better. And we call these research ambassadors. And every month we get together on a big teleconference and we connect the research ambassadors with different stakeholders. And we give them opportunities to influence research. And that could be, raising money so that we can do more research, changing laws, so that we can do more research, optimizing the questions that we're asking, the designs of studies, spreading the word about new studies, and helping regulators to understand patient perspectives. I'll leave you with this. There's 2 quotes that I've come across that constantly remind me of the importance of patient engagement. The first one is a patient who told me, "Nothing about us should ever be done without us." And I've tried to embrace that with everything I do. Whenever I participate in a meeting, I want to make sure that there are patients invited. I think it's so important to have patients at our meetings. And when I do studies, I want to make sure I'm bouncing the protocol off educated patients to help me understand if there's any way I can make it more patient-centric. And the other quote that sticks with me is "better together." I think there's not nearly enough people in the world that care about ALS. We cannot afford to be fighting with each other. We've got to find a way to work together to find the cure for this disease. Another thing that's happening in ALS research that I'm very excited about is biomarkers. So, we've had very clumsy ways to measure ALS. Basically, they're symptom-based scales like the ALS Functional Rating Scale, or they're exam-based scales like manual muscle testing or forced vital capacity. And now for the first time, we're on the cusp of having biomarkers. And biomarker is just a way to measure something. So we have digital biomarkers. For example, we've got an app now where people can actually talk into an app on their phone, and it will analyze the rate and intelligibility of their speech. We've got blood and spinal fluid markers, like things called neurofilaments, that turn out to be probably much more sensitive and specific markers of motor neuron health than the clinical measures we've been using. And we've got markers that tell us about those different downstream pathways. It turns out that some of those pathways might be much more important in some patients than in others. And so what I've shown you here is a PET ligand, something that we can inject into people, that is picked up by activated microglia. And so down here, we've got the average of what 10 brains who are healthy look like on a PET scan. And we've got the average of what 10 brains from people with ALS look like when we inject them with this PET ligand. And you can see that there's these hotspots in different parts of the brain. And so, if we were doing a clinical trial of a drug like verdiperstat that might block the activation of microglia, you could see how important this could be. We wouldn't want to give that drug to people who didn't have activated microglia in their brain, and it might only be a subset of people with ALS. So I think these markers are going to revolutionize the way we do trials. We're going to be able to enroll the right patients, and we're going to be able to follow things that change much more quickly than the clinical measures that we've been using. Platform trials are very exciting. This is another revolution in how we do research. And it seems so obvious now, but it wasn't. We got this idea from cancer biologists. We used to build an entire trial infrastructure every time that we wanted to study something new. So we would write a new protocol. We would figure out who we needed to do what in the protocol. And we would have to get lawyers involved and get those people under contract. We would have to take the protocol to the FDA and get their permission. We would then have to take the protocol to our institution and get IRB and departmental approval. And this would all take, on the order of 6 to 12 months to get all this to happen. And then we would use all that and we would study one drug and we would smash the whole thing down and build it all back again to study the next drug. But the platforms trial says, why don't we just build it one time and call that the master protocol and plug new drugs that we want to study into the same protocol and studying in the same way. And so it's much more efficient way to do research. And each new drug that we plug in has a 3-to-1 ratio with a placebo. And we can get away with that because we can pool the placebo groups across all the different arms of this study. And as a result, this means that more patients get to try experimental drugs. And as you probably know, the first ALS platform trial opened in July of 2020 at MGH, and it's now starting to open at many centers across the United States. Virtual visits are another thing that's happening in our trials. I mentioned this earlier. I think some of us were trying to make this happen for a long time, but the COVID-19 pandemic really catalyzed it. Most trials now offer virtual visits in place of in-person visits. Now, not all trials can be entirely virtual. There are some trials that require us to measure things in person, like, for example, some trials that require us to do things that we can't yet do virtually. But there are some trials, pilot trials, that are becoming entirely virtual, and that's my Therakerman trial at Duke, and it's also the NIH HERV-K suppression trial that I told you about, which can be done entirely virtually. Historical controls, this is still controversial, but I am personally one who believes that we now have ways to generate historical controls. Placebo controls are still what regulators want to see, but I think that we have ways to model the progression of people with ALS and use this information so that we could treat everybody in a pilot trial, at least, with a drug and figure out if they were doing better than they were predicted to do. So what I've shown you here is the results of my Lunison trial. In this trial, we took the progression rate of every one of the 50 patients, and we found someone who was progressing at a similar rate in the Patients Like Me database. In fact, we actually found 3 people progressing at the same rate. So each person in the Lunison trial had 3 virtual controls from the Patients Like Me database. And we looked at how our patients did on treatment over 12 months compared to how they were supposed to do according to their historical controls. And what you can see here is that we almost perfectly predicted the ALS FRS progression of these 50 patients over 12 months. So the bad news is this drug, Lunison, had no effect whatsoever. But the good news is this method of predicting progression appears to work extremely well. And I feel like we can now use things like this to do at least pilot trials without placebos. Expanding access. Almost every person I've ever met with this disease wants to try experimental treatments. But as I said before, not all can participate in clinical trials. And so there do exist pathways for people to get some of the exciting products that are being studied outside of a trial. Historically, these types of pathways, which are called pre-approval access pathways, in ALS have been rare. And so a couple of years ago, as most of you know, something happened called Right to Try. And we all hoped that this was going to change everything, that people with ALS who couldn't get into a trial would now be able to access these exciting products. Unfortunately, this law turns out to have been mistargeted. So there are 4 things that have to happen for a person with a disease like ALS to get access to one of these products that's in development. First, they got to ask for it, and they almost all do, in my experience. Second, their doctor has to be willing to support their getting it. Some doctors are and some aren't. Third, the company who owns the product has to be willing to give it out outside the trial. Some companies are and some are not. And finally, the FDA has to be willing to field an application from the doctor and the patient that explains exactly how they're going to administer the drug and monitor the patient for safety. Now, it turns out that that's the easiest of the 4 things. Very simple to fill out this application and get it approved by the FDA. And the FDA has actually approved almost 100% of all these expanded access applications that it's gotten over several decades in fields like cancer and HIV. So Right to Try actually addressed the FDA and said, we're going to move them out of the equation. It's only up to these first 3 stakeholders. Since that was never the problem in the first place, unfortunately, Right to Try really didn't do anything. There really aren't a significant number of people with ALS who got to try anything as a result of the Right to Try. And that's because the barriers, again, are the physician. Many physicians are not confident about these expanded access programs. Many physicians do not have the resources at their site to participate in these. Many physicians are stretched as far as their time as it is. And the companies. Many companies are fearful of these programs. Many companies may not have the resources. If you're a small company and you've only made enough of the drug to do your clinical trial, how could you possibly have enough to potentially field 20,000 expanded access applications? It doesn't make sense. So the good news is we're starting to see some progress in terms of actually targeting the true barriers to expanded access. So first, there's this awesome grant that IMALS put together, and it was matched by the company Biohaven. And this is going to allow us to offer expanded access to initially 35, eventually 100 people who can't get into the Healy platform trial. It's also going to be used to create new materials that we can eventually use to educate physicians who want to be able to provide EAPs, but maybe don't quite understand how to do it. So we'll create training and advocacy materials and an expanded access training course at our NEALS meeting with some of the funds from this grant. You may also know that there are some bills in Congress that will really help with these barriers to expanded access. And so among other things, these are going to create a federal fund that could be used by investigators or by industry who want to be able to offer EAPs and study their outcomes. So the last thing I'm going to touch on is something that, again, is really hot off the press. In fact, really just sort of solidified this this afternoon. But this is called high throughput drug screening. So if you haven't heard this before, high throughput screening is a proven way to accelerate drug development. It's used all the time in basic science and even in preclinical animal labs. And the idea is this. Let's say that next year there's a new bacteria that is resulting in another pandemic. Hopefully that's not going to be the case, but just indulge me for a second. If I wanted to figure out which of all the antibiotics we have available to us, all the things that are FDA approved, could kill that bacteria, what I could do is I could put a little bit of that bacteria in all these little tiny tubes, and I could put this into a machine and ask it to squirt every FDA approved antibiotic into one of these wells. And I could quickly see which ones affect this bacteria. And taking this to the animal level, we could build mouse models of ALS, and we could screen large numbers of drugs in these models to see, are there any FDA approved drugs that seem to actually help these animals to live longer with their form of ALS. And in fact, that's part of what ALS TDI does. They do high throughput drug screening in animal models of ALS. But nobody does this in people. Why? Well, I suspect I know the answer, and it has to do with what I told you before. Each experiment that you might want to do with a patient of a new therapy, you'd have to write a protocol, and you'd have to get the people that you needed to help you under contract, and you'd have to get that approved by the FDA, and you'd have to get that approved by your institutional IRB and by your department, and it would take 6 to 12 months for each N of 1 experiment. The thing is, though, this is already happening. It's just that nobody's recording it, right? I told you before that people with ALS are experimenting with things. In rare cases, expanded access. Hopefully that's going to be more often the case, but just about everybody I've ever seen with this disease has their own regimen of alternative and off-label treatments that they're experimenting with. Some people are just on one thing, and some people are on more than 50. But nobody is actually paying attention to all these thousands, potentially hundreds of thousands of N of 1 experiments that are going on around the world. And so what patients like me has agreed to do, which I think is really cool, is create something called a guided experience, where you'll be able to go in there and tell them a little bit about your disease and tell them what things you've decided to take. And they will generate historical controls for you so that you will get an idea of how you are expected to progress on the ALS FRSR score over the next 6 months. And you can see if the regimen that you've selected is allowing you to do much better than you were expected to do. If not, well, then maybe you might want to change your regimen. But the other thing is, we're going to be able to get an alert if we see that there are certain regimens associated with a dramatic amount of slowing, stopping, or reversing of the expected progression of the disease. This will immediately tell us about new treatment candidates that we might want to take into more traditional clinical trials. So I call this high throughput drug screening in people with ALS, and I'm very excited about it. So what I've shown you in the past hour is that in the years that I've been involved in ALS, we've made some progress. We now have a lot of different treatment options that it can improve quality of life, and combinations of these that are addressing downstream causes of ALS, downstream events that are killing motor neurons, are incrementally slowing progression. But for the first time in history, we now understand some of the causes of ALS. And targeting those, I think the sky's the limit in terms of what we may be able to do. And we're also starting to understand, or at least we have plans to understand, differences in people with ALS in terms of how they progress. And that's going to lead to exciting new treatment candidates. And once we have these candidates, improved patient engagement, platform designs, trials with virtual visits and historical controls are going to allow us to test these things more efficiently than we ever have before. And finally, we are soon going to be able to study larger numbers of people with ALS on experimental products than ever before through expanded access and through this tool on patients like me. And I personally believe this is going to speed our search for better treatments. And so there's lots of people that I need to thank. Most importantly, those affected by ALS who are on the call. You constantly inspire me to work harder. I mean, you're the main reason that I'm still in this field after all these years. I want to especially thank the research ambassadors and those who participate in all the amazing research and advocacy. We would make no progress without cooperation from you. All the funding sources for my work and all my collaborators. And finally, I want to thank, the people who are on my Duke ALS care team for helping me to build a foundation of great patient care here, upon which all these ideas for research are built. And so that's the end of my talk. I will now open up the floor for questions. I think I have 10 minutes. Is that right, Nicole?
Yes, it is. All right. We do have some questions in. So let me begin. What is the average dose reached in the Clinbuterol group? Was there a common dose for the dropouts?
It's a great question. I don't have that. I don't have that information yet, but it's part of the things that we're analyzing is to figure out. We allowed in the protocol for people to titrate all the way down to a small starting dose, but even that was higher than what was being used in that previous study. So if I were to take Clinbuterol forward, which I hope I can find partners that help me, I will probably use the dose that was used in the previous trial, which seemed to have a pretty good effect and much better tolerability than the doses we used.
Okay. This is an interesting, this kind of is a statement and a question. Long shot may be a coincidence. 3 weeks ago after my Moderna vaccine, I had improvement in function. Almost impossible to roll over in bed, to rolling over with ease, needing to use my arms to push me up from sitting to standing, and now I can stand without the assist of my arms. Have you heard of anyone else experiencing this after their COVID vaccine?
I haven't, but that is fascinating. And I will tell you that at the Charlotte ALS clinic, the Carolinas ALS clinic in Charlotte, they are leading a COVID registry. And so they're trying to get as many people with ALS who had COVID to enroll in their registry. And they also want to know about people with ALS who had the COVID vaccines for exactly this reason. And, I think one of the things that amazed me is there is some data to suggest that people with ALS were especially at risk from COVID-19.
Mm-hmm.
Like I saw 2 of my own patients who were hardly changing over years and years of follow-up who rapidly progressed after they got COVID-19. And there's a researcher at the VA who's been able to look at a very large database of people with ALS who are veterans who got COVID. And the mortality, in the 30 days after COVID is much, much higher than expected. It's like around 15% as opposed to, 1 to 2%, which is what we would expect for people without ALS. So I think, we really need that kind of registry to understand this better, but we also want to capture, potentially good and bad outcomes from the vaccine in that registry. So I'd encourage you to look up the Carolinas ALS Center in Charlotte and reach out to them and let them know that you'd love to sign up for their registry.
Okay. Does Riluzole continue to be effective after 6 months or so, or does it stop working after a time?
Yeah, it's a great question. So, early in my career, I think most of us thought there was a point in time where Riluzole probably didn't make any difference. And then a couple of years ago, we saw some reanalyses of the pivotal Riluzole trials that suggested that it works at every stage of the disease. Even the latest stages of the disease that we can measure, it still seems to be slowing the disease down. So that changed my practice. I mean, I for patients who are on Riluzole, I continue the Riluzole unless they tell me they've gotten to a point in the course of the disease where they just don't want to, prolong it anymore. But that's not most of my patients. Most of my patients want to keep fighting all the way through the disease.
Yeah. Okay. Is there any evidence that trauma, if you experienced a trauma, that could have caused ALS in your body?
Yeah. So the bottom line is that some studies suggest that could be a trigger and other studies don't.
Okay.
The best meta-analyses haven't found a consistent effect. But this goes back to what I was saying before. It's almost certain that there isn't one cause of ALS. There's not one genetic cause. There's almost certainly not one sporadic cause. And in fact, most of us believe that in order to get ALS, you probably need multiple things to happen to you over the course of your life. And that combination of things is probably different in different patients.
Mm-hmm.
So let's say that there's a collection of genes that make you susceptible to trauma. If you never get trauma, you never get ALS. And let's say you don't have that combination and you get several severe traumas. Well, then you don't get ALS either. But if you have the combination of susceptibility genes and you get the trauma, then you get ALS. I mean, it's a theory, but I think it's got to be right. I mean, I've just seen too many cases over my 21 years of people whose disease appeared to start after a surgery or, a physical trauma. I
Okay.
believe there is a subset that is triggered that way. It's just really hard to figure out how to identify that scientifically because it's not everybody, it's just a subset.
Okay. Is there any connection between mental health issues and ALS, particularly because of the mind-gut connection and your findings about the microbiome?
Yeah.
And Dr. Catherine Loman-Hurth is going to be on tomorrow. She's going to talk a little bit about that as well, but do you have any comment to that?
Yeah, so Kathy is really one of the world experts in this, but it turns out that surprisingly, about 50% of people with ALS have cognitive and or behavioral problems when you screen them carefully. Now, when I first heard about this, I said, that can't be right, because even though I wasn't doing formal screening, I was, of course, spending, 2 hours with every one of my new patients talking to them. And it just seemed to me like it was more like 5%.
Mm-hmm.
But since I started screening, I found out that she's absolutely right. It is more like 50%. And even though these problems can be subtle, they're really important because people who have these subtle problems tend not to make good decisions about the options they choose. They also tend not to be compliant with the options they choose. So for example, non-invasive ventilation. I often find that my patients with cognitive impairment are not using it for at least 4 hours a day. And so they're not reaping the huge survival benefit that goes along with non-invasive ventilation. And it's been shown over and over again, including by Kathy's group, that if you have these problems and you're non-compliant, you don't do as well. You're more likely to be hospitalized and you don't live as long. And so when we find these problems in our patients, we try to empower the family to take a greater role in decision-making and monitoring compliance.
Okay. Is tofersen being evaluated as part of a platform trial?
No, it's currently not, it's not part of the platform trial. It's in its own separate trial. And, hopefully that second trial will look as good or better than the first small pilot trial, because I think if it does, it will probably get approved. So there wouldn't need to be any other trials.
I'm sorry, my doorbell is ringing right now. Let me ask, she also wanted to, this was a second part to her question. Why are patients unable to gain access to therapy under compassionate use? She would like to understand that a little bit better.
Yeah, so the term compassionate use, it means the same thing as expanded access. So there's actually multiple types of expanded access programs. There's single patient expanded access. That's where one person comes to me and says, I want to try this drug. Will you help me? And I say, yes, let's reach out to the company. Let's see, if the company will make it available to you, what the cost will be. Let me figure out if I can get the resources. And if all that's yes, 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 then we put together a short application. We send it off to the FDA. We say, we want to give this to this one person with this terrible disease. So single patient expanded access is the same as compassionate use. There's also intermediate expanded access, which is a small group of people. That's what's going to happen in the Healy platform trial. And then there's large group expanded access, which is what I would really like to see, where it's more than 100 people who are getting access to the product outside the trial. And there you could actually start to gather some useful information about, is the safety signal different? Is it more effective or less effective in this group of patients compared to who's in the trial? But when it comes to even the single patient expanded access, the main reasons why there's not more of it are, number one, doctors don't understand it. They're uncomfortable with it. Many of us don't have the kind of resources that we really need to support these programs. And hopefully all that's about to change. But the other problem is that most companies have not been willing to give their drug out even to a single patient outside of a clinical trial. So it doesn't matter if there's a law passed called Right to Try that sounds really great. If the person that owns the product is not going to give it out, the law is meaningless. So we need to find ways to educate companies, to help companies who want to do this but don't have the resources. And that's what some of these grants and some of these laws that are in front of Congress are designed to do. So I think this is about to change big time, which I'm excited about. But that's why traditionally it's been so difficult.
Sorry, this person was commenting that tofersen would be a great alternative, but its price is a problem for patients to access. especially with those in the health systems of Latin American countries. Um, do you know, or could you speak to any way about how there could be a resolution to that issue of a high
Yeah.
cost?
So the first thing I'll say is that sometimes the cost of things is a lot less than we expect. So I was on a call maybe a week or 10 days ago, and someone said, well, Riluzole is just so expensive. And it turns out if you go on the website GoodRx, you can get Riluzole for $50 at most pharmacies, okay? Even with no insurance. And the other thing is that there's a group called the Health Well Foundation. If $50 is a lot for you, they will reimburse you the $50 for the Riluzole for your ALS. So the net effect is for people who have trouble affording Riluzole, it can be free. Now,
What was that
so the
company
first
again?
one is called GoodRx, OODRX, which is where I always go when I get prescribed a drug by my doctor. I go on there first, see, oh my God, this thing is $300 cheaper right across the street. It's amazing. And then the other is Health Well Foundation, which helps people with ALS with currently FDA-approved drugs, including Riluzole, Radicava, and Nudexta. It helps with the cost of all those. Now, tofersen, right now, it's not approved for anything. So, you know, when you say it's expensive, well, we have no idea what it's going to cost. It may very well be expensive because, you know, it's quite an unusual drug. It's also not a pill. It's going to have to be infused into the spinal fluid, possibly as often as once a month. So there'll be a cost to the drug. There'll be a cost for that procedure, for the physician time, all that stuff. So it could be expensive, but at this time we don't know. Hopefully what will happen with this new drug, which happens with a lot of drugs, is that companies recognize that, you know, people all around the world want to get this, and they come up with patient assistance programs. So hopefully Biogen, if this looks as great as it did in the first study, will have a patient assistance program that will allow people who can't access it, you know, through their insurance to get it that way.
Okay. Could you provide the link to Patients Like Me throughput site?
Yeah. So right now, what I was telling you about doesn't exist. The link to Patients Like Me, you can sign up for Patients Like Me right now. It's just patientslikeme.com. But the guided experience that we talked about is being built. And so I just today got the double thumbs up from them that yes, we're definitely doing this. But that tool, I think, is going to be so much more powerful. Patients Like Me is great for a lot of reasons. I partner with them on my virtual trials. That's the database for my virtual trials. And you can go on there right now and meet other patients who have ALS, meet other patients who are similar in age and onset and speed of progression. You can compare, you know, your own impressions of the products that you're using with other patients. But I think this new tool is really going to empower patients in a way that we never have been able to before, to determine for themselves, should I stay on this regimen or should I switch to something else? Not all doctors will do that with patients. Also, you know, I am so curious because for the first time now I'm going to have like some official records of all the thousands of individual N of 1 experiments that I know are going on around the world, some of which may be working that I don't know about. So I'm so excited to see that for the first time.
That will be very exciting. Someone has typed in, are B12 injections helpful?
Yeah, so there has been some research from Japan suggesting that a B12 precursor called methylcobalamin might slow the progression of a subset of people with ALS. Now, interestingly, this subset is patients who have symptoms for less than a year. I almost never see that subset of patients in my clinic. Unfortunately, this problem, this so-called diagnostic delay, is something we didn't talk about today, but it is a huge issue. There often are a period of months that go by before a person with ALS ever sees any doctor because they're not sure anything's wrong. The disease starts gradually and it sneaks up. And then the first doctor is typically a primary care doctor. Primary care doctors think about common things, which ALS is not common. It's not as common as a pinched nerve. So oftentimes there's anti-inflammatories and physical therapy and a referral to a surgeon. And then finally, you know, maybe 6-8 months into this, they see a neurologist who recognizes right away that this is something much more serious than a pinched nerve, often writes, you know, possible motor neuron disease in their notes, doesn't break the news. And I think there's probably two reasons for that. First, they may not be confident since we don't have a diagnostic test. It's a clinical diagnosis. They may not be confident and want to break that to someone without being sure. And second, it takes a lot of time. When you put those words out there, you can't just, put those words out there and walk out. I mean, you got to explain what that means, the variability, the things we have to offer, the hope. That's why my new patient visits are typically 90 to 120 minutes. It's not because I'm a slow examiner. It's because I take the time to try to make sure people understand this. And, if you're in private practice, you cannot take that long with one patient. You might have 10 minutes scheduled for that patient. There's no way you can break that news in 10 minutes. So they refer to an expert like myself who specializes in this. And there's just not enough of us. There's typically a few month wait to get into our clinics. And that's why it's about a year or more, when I see someone for the first time. So by the time I see them, there's no data that methylcobalamin helps after it's been going on for a year.
Okay. Would you say ALS can be diagnosed by eliminating all other possibilities?
Yes, I think that's part of it. You have to first confirm that someone has a progressive motor neuron disease with upper and lower motor neuron involvement. And then the second part of it is excluding mimickers. So that's all the stuff that's wrapped up in making a diagnosis.
Okay. How would someone contact you about your reversal group?
Yeah, so my email address is richard.bedlack@duke.edu. And we do have a website, which is www.alsreversals.com. You can see some of our references that we've published on this. You can see some of our protocols that we've done and that are currently open.
Good. Okay. This person says their mom was initially diagnosed with bulbar palsy. How is this related to ALS? Because now her doctor is calling her condition ALS.
Yeah. So just like there's differences in the rate of progression, there's differences in the way this disease starts. So a person who has only upper motor neuron signs in their bulbar region, no upper motor neuron signs anywhere else, no lower motor neuron signs anywhere else, is said to have bulbar palsy or pseudobulbar palsy. Okay. Now, unfortunately, most of these motor neuron disease variants do progress into full-blown ALS. So, some people will start with stiffness in their legs, upper motor neuron signs in their legs. And at first, doctors might say, well, this could be PLS, primary lateral sclerosis. Unfortunately, in my experience, most people who present that way will eventually progress into full-blown ALS. We just have to follow them long enough. And the problem with this is that all those delays matter. The delays matter in terms of how effective our treatments are. They matter in terms of getting into a clinical trial. Somebody that I just saw this week, they've had symptoms for 2.5 years. They're already excluded from a lot of clinical trials because they've had symptoms for more than 2 years. So it's not an easy problem, but we have got to find a way to get people diagnosed and under the care of experts faster.
I definitely agree with that. My brother only got an EMG and was diagnosed. Nothing else was done. No MRI or CT scan. Should one of those tests been done as well before they got that diagnosis?
I mean, it really depends on how someone presents, right? So if someone presents and they only have one body part that's affected, well, there's a long list of possible mimics of that. But if someone presents and they have upper and lower motor neuron findings in their bulbar region, in their arms, in their legs, in the middle of their back, I cannot think of any sort of structural problem that could cause that. Like you can't push on every motor neuron at every level of your brain stem and your spine. So it really just depends on how someone presents. And, in my experience, there's almost always mimics to exclude. And two very rare, but potentially reversible ALS mimics that I'm always careful to exclude: copper deficiency. So I make sure all my patients have a blood copper level, and celiac disease. And so I make sure that we do a celiac screen. And if that comes back abnormal, then we do an intestinal biopsy. Because even though they're extremely rare, they're potentially reversible. Yeah.
And I just want to be cognizant of our time. Just a couple more questions. Why do vets have twice the risk of getting ALS?
I wish I knew. I've got a couple theories. So, one theory is that it's physical fitness. There have been multiple studies that have identified physical fitness as a risk factor for ALS. For example, if you've ever played a varsity sport at any level, one study suggests that you're about twice as likely to get ALS as if you never played a varsity sport. And if you think back through history, military veterans on average were more physically fit than other people from their same generation who didn't go into the military, especially when we had a draft.
Exactly.
In the draft days, if you got drafted and you were in horrible physical condition, you didn't get into the military. So I think that's one possibility. The other possibility that I've actually heard from the veterans themselves, which I think is a fascinating idea, is that military veterans, no matter when or where they served, often get a large number of vaccines in a short period of time, which is not true to my knowledge of just about any other occupation.
That's very true, yeah.
Yeah, some of them tell me, in basic training, they kind of walk down this line and people were just injecting them, with all these things.
Yeah, yeah.
So I don't know if you have some susceptibility genes and you get a massive activation of your immune system over a short period of time like that, if it's possible that could trigger the disease. I think that's a fascinating theory. It's hard to figure out how to study that. Like, it would be great if there was another group of people that we could identify who got a whole bunch of vaccines over a short period of time like that, because we could look to see, is there an increased risk of ALS in that group too? But I don't know of one.
Yeah, that would be interesting. And the last question, and then we'll wrap it up and get to our drug development panel. Do all 52 of your reversals that you've seen have anything common within that group besides the reversal?
Yeah, the short answer is not that I have figured out yet.
Okay.
So I have a lot of ideas, a lot of collaborations underway to try to look for commonalities in genetic polymorphisms, RNA expression, protein biomarkers, microbiome, environmental exposures, everything that I can think of to test in these people and compare it to people with ALS that doesn't get better, I'm testing. So, we'll see. Okay. But I think over the next year, a lot of the results of these things will be in, and I'll have a lot more information.
Well, we will be waiting for sure. I want to thank you very much for your time today. And I know you're going to stick around and you're going to moderate our panel. Sure. Thank you.